video, we're going to cover Chapter 3 and the first part of Chapter 21, just pages 603 to 611, and we're going to be looking at cell metabolism. So in this first part, should just be review. We're going to take a look at the types of metabolic reactions and also talk about energy. So just a little bit of terminology. So when you're looking at a chemical reaction, remember that what you start with over here on the left, these are the reactants. And then over here on the right, what you end up with when you're finished, these are the products. Okay, just a couple of definitions. Metabolism. These are just all of the chemical reactions that are going on in a cell. And these include both anabolic and catabolic reactions. So remember, anabolic reactions are when you're taking smaller molecules and putting them together to make a larger molecule. So maybe, for example, you're taking amino acids and joining them together to make a protein. So that would be an anabolic reaction. And remember that anabolic reactions are endergonic, because they require energy to happen. You've got to put energy in, and that's what endergonic is referring to. So they require energy. Catabolic reactions are just the opposite. So you start with a larger molecule and you break it into smaller molecules. So for example, maybe you've got glycogen and you break it down into glucose molecules. So that would be a catabolic reaction. And catabolic reactions are exergonic, and that's because they release energy. So when you're breaking apart that larger molecule, breaking apart those chemical bonds, you're releasing energy. So the way that I remember this is I remember anabolic steroids like bodybuilders take, and I remember that they get bigger. So that's how I remember that anabolic reactions, you're taking smaller molecules and putting them together to make a larger molecule. And then catabolic reactions, I remember because I think of cats, especially if you have a young cat, they can be pretty destructive and shred things up. And so they'll take something larger and shred it into small pieces. So here's just a really nice overview of what's happening in your body with these catabolic and anabolic reactions. So we've got some food that you've eaten. Maybe you've had some Lucky Charms for breakfast here. And in your digestive tract, you're going to do some catabolic reactions and break down your food into smaller molecules so you can absorb those. And then once those get into your cells will then do some anabolic reactions and take those smaller molecules, maybe take some amino acids and put those together to make some proteins, for example. So that's just kind of showing you how these catabolic and anabolic pathways are going to be used in our body. The other thing to notice is that you have to take in more food than what you're going to be able to make in the body, and that's because you're always going to lose some of the energy from your food as heat. And that's that second law of thermodynamics, because you're not going to be 100% efficient at converting energy from one form to the next. You're always going to lose some energy as heat. Okay, so this just shows exergonic reactions. So the reactants have more energy to start with, and when you break them apart, break the chemical bonds, holding that larger molecule together, we're going to release some energy, and then the products end up with less energy down here. And if you remember, the delta G, the free energy, ends up being a negative number, just indicating that you're releasing energy and there's energy available to do work with. So these exergonic reactions release energy. Also notice that they're going downhill. And so these are going to be catabolic reactions. We also call them spontaneous reactions, just meaning that they're energetically favorable. It doesn't mean that they happen right away, but it just means that they're energetically favorable. And then endergonic reactions, we've got to put energy in to go up the hill. So the reactants have less energy to start with than the products that you end up with at the end. So maybe you've got some glucose molecules and you're going to join them together to make a large glycogen molecule. So you've got to put energy in to make all those chemical bonds. And so your delta G ends up being positive, indicating you've got to put energy in to go up that hill. And these anabolic reactions are going to be endergonic, just saying you've got to put energy in to do this to go up the hill. And we also say that they're non-spontaneous, just saying that they're not energetically favorable, just saying, hey, you've got to put some energy in to go up the hill. So when we do catabolic and anabolic reactions in the cell, a lot of times they may not be occurring at the same time or maybe even in the same place in the cell. So we need somewhere that we can temporarily store the energy released from catabolic reactions until we can use them in the anabolic reactions. So we're going to use ATP to do that. So here's a little picture of this. So if we've got maybe some food here that we're going to be breaking down, we're doing our catabolic reaction and we're going to be releasing some energy here, and then maybe we're going to be making a protein over here. And so we're just going to take that energy and then use that energy released from our catabolic reaction over here and we'll use that energy to phosphorylate or put the phosphate group onto ADP to make some ATP, and then we'll use the ATP to provide energy maybe at a different time or a different location in the cell to do this anabolic reaction. So maybe we're taking some amino acids and making a protein over here. And then this is just what ATP looks like. So if you recognize the structure of this, it has 3 parts. So you can see that there's a 5-carbon pentose sugar here. This is a ribose sugar. And then we've got a nitrogenous base, which is an A. And then we've got 3 phosphate groups. So ATP is actually an RNA nucleotide. So if you're looking in RNA and you see the A's in there, that's actually ATP that's in there. So, okay, and where the energy is stored in ATP is in these covalent bonds that are joining the phosphate groups together. So that's where all the energy is stored. Okay, and then this just shows how ATP links these catabolic and anabolic reactions together. So if you're taking some of your food that you've eaten and you break it down, release the energy stored in the food, you're going to temporarily store that in ATP. And then, maybe another time or a different place in the cell, you can chop off one of the phosphate groups on the ATP, release the energy there, and use that to do one of the anabolic reactions, maybe make a protein or something. And then we can just reuse that ADP and then remake the ATP again when we eat more food. This is a nice summary of what's going on in the body with ATP. So we've got some food, some glucose here, and we're going to break apart all the bonds in the glucose. And here's our waste product, carbon dioxide, that we'll exhale. And then we're going to release that energy from the glucose and temporarily store that in ATP. Although not all the energy in the glucose goes into ATP, we do lose some of it as body heat. And then the ATP, we can chop off the phosphate group from that and then use that energy from the ATP to do work in the cell. And there are 3 different types of work that the cell will do. So there's chemical work, and the example there would be anabolic reactions. So maybe you're taking amino acids and making a protein. And then we have mechanical work. So that would be, for example, maybe contracting muscles. And then the last type of work would be transport work. So that would be any type of active transport. So maybe you're doing primary active transport or secondary active transport or endocytosis or something like that. So that's what the cell does with all the ATP that you produce from the food that you eat. Okay, we've got a couple of different types of chemical reactions just to remind yourself of. So we've got condensation and hydrolysis reactions. They're the opposite of each other. So if we start over here on the left and go over here to the right, you can see we've got 2 monomers, 2 little subunits. And then what we're going to do is we're going to join these monomers together and squeeze a water molecule out and form this dimer. And di just meaning 2 and mer means part. So we've got 2 monomers joined together to make this dimer. So going from the left to the right, that would be a condensation reaction. And the reason they call it that is because you end up squeezing a water molecule out, and it's just kind of like when you take a shower and the water condenses on the mirror, you end up producing that water. So that's where the name came from. Okay, but if you start over here on the right, so you've got this larger dimer, and then you end up breaking it apart into the 2 monomers, that would be a hydrolysis reaction. Okay, hydro talking about water, lysis is to split or break apart. And notice what you're doing is you're adding the water and then breaking it into 2 smaller pieces. Okay, and there is an enzyme involved here that's catalyzing this reaction. You know, not just adding the water is going to break the bond, you've got to have an enzyme as well. Okay, so if we take a look at this, if we go from the left, take 2 smaller pieces, join them together to make a larger molecule, the condensation reaction is an anabolic reaction. And then if we start over here on the right, and then go to the left, do the reverse reaction, do this hydrolysis reaction, we have the larger dimer, split it into 2 smaller pieces. The hydrolysis reaction will be a catabolic reaction. So condensation reaction is anabolic, and hydrolysis reaction is catabolic. Okay, we also have oxidation reduction reactions, and the way you remember this is oil rig. Okay, so oil stands for oxidation is loss of electrons or hydrogen atoms, and rig stands for reduction is gain of electrons or hydrogen atoms. So when you're trying to figure this out, it'll always ask which reactant is oxidized and which reactant is reduced. So remember, the reactants are what you start with. So if you look over here on the left, this is what you start with. You've got glucose and oxygen. So those are the only 2 that you've got to choose from. Okay, and then you need to look over here on the product side to see, well, what happened to them. And whatever is written first on the left will be written first on the right. So glucose becomes carbon dioxide, And then oxygen being written second will be written second over here on the right, so oxygen becomes water. Okay, so if we look at glucose, notice the hydrogen atoms. We've got lots of hydrogens in glucose. And then if you look over here on the other side, by the time you get to carbon dioxide, they're gone. So we would say that the glucose, that reactant, is oxidized because it lost the hydrogen atoms or electrons. So that's the oil part. So glucose is oxidized because oxidation is loss of hydrogen atoms or electrons, because you go from lots of hydrogen atoms to nothing over here on the carbon dioxide. Okay, so the glucose is oxidized. And then if somebody's losing hydrogen atoms or electrons, well, somebody's got to gain them. So if you look at the other reactant, the oxygen, and see what happens to it on the other side, it turns into water. And so you can see that the oxygen has gained hydrogen atoms or electrons. So that's the rig part. So reduction is gain of hydrogen atoms or electrons. So we would say the oxygen, that reactant, oxygen, is reduced because it gains hydrogen atoms or electrons because it turns into water. Okay, and then the last type of chemical reactions to talk about are phosphorylation and dephosphorylation reactions. So with phosphorylation reactions, we're going to add a phosphate group on. So maybe you've got a protein here, and we're just going to make a covalent bond and stick a phosphate group onto that protein. So that would be a phosphorylation reaction. A dephosphorylation reaction, D means to remove. So a dephosphorylation reaction, what happens there is you have this phosphorylated protein, it has a phosphate group on it, and what happens is you remove that phosphate group. So you chop it off. So that would be a dephosphorylation reaction. Okay, the law of mass action, we talked about this a little bit in chapter 2. So by changing the concentration of the reactant or the product, we can change the direction of the reaction, which way it'll go. All right, so let's say that we increase the concentration of one of the reactants. Let's say we increase the amount of X. So if we put a lot of X over here, this reaction is going to say, hey, now all of a sudden I've got way too much X. So we need to get rid of X. So the way to get rid of X is if you go to the right and you end up over here at Z, and then there's no more X. Okay, so if you increase X, that's going to push the reaction to the right. Okay. And then if you increase the amount of the product, let's say you add a bunch of Z. Okay, so if you add a bunch of Z, the reaction says, hey, wait a minute, I've got way too much Z now. We need to get rid of Z. So the way the reaction does that, it says, okay, let's go the reverse direction and let's go to the left. Because if we end up over here on the left, the Z is gone. We get rid of some of it. Okay, so if you increase the concentration of the reactants, so either X or Y, that would push the reaction, will go to the right. And then if you increase the amount of the product, Z, the reaction would go to the left. Okay, because you're just trying to get things back to where they were before you added too much of something. So that's the law of mass action. Okay, the activation energy, I know you've learned about that before. So that's the amount of energy you've got to put in to get the reaction started. So maybe the molecules have to collide with enough energy to get things started, or maybe you're going to put enough energy in to start breaking the bonds. So the activation energy is just the energy of the transition state minus the energy of the reactants or the product. This activation energy is actually really important because it limits how fast a reaction can go. Okay, so let's take a look at a picture of this. All right, so we've got two different reactions here. So let's say that we're going to go to the right, and we're going to end up going down the hill here. So our activation energy for going to the right here is this little hill here. Okay, that's our activation energy. So that would be our E sub A. Okay, so that's the amount of energy we have to put in to get the reaction started. So we got to give things, it's like you have a rock sitting at the top of the hill and you got to give it a little shove to get it rolling down the hill. Okay, so that's not very much, and so that reaction, you know, is pretty likely to go because it doesn't take much energy to get it going. But what if we were doing the reverse reaction? What if we started over here on the right and we want to go to the left? Well, look how big your activation energy is over here. You have to put in from down here all the way up to the very top there, and that would be your activation energy if you were going that reverse direction. So that reaction going to the left is not going to happen very fast because it takes a lot more energy to get that reaction going. So that's going to be a very slowly happening reaction than the one going to the right. Okay, and there's a couple of factors that affect how fast a chemical reaction happens. The two that are most important in the cell are the concentrations of the reactant and the product, so the law of mass action, because cells can change that. They can add or subtract the amount of reactant and product, and that can shift which way the reaction is going. And then also, cells have enzymes, and that will catalyze or speed up the rate of the reaction because that'll lower that activation energy barrier. Cells really don't change the temperature because hopefully we're maintaining homeostasis with our body temperature because we don't want to be messing with that. But obviously temperature can affect reaction rate if it gets too hot or too cold. That can affect the speed of the molecules moving and affect the reaction rates. And then we talked about the height of that activation energy barrier. So the higher it is, like if you were doing this reverse reaction and you had to go from way down here to way up there, that's going to be a really slow reaction. And if you had the forward reaction with just this really small activation energy barrier.